So the GDP is inactive, correct?
GDP is an inactive, lower energy form.
Okay, and then it turns into GTP.
It goes back and forth. So it has to have a phosphate group added to it. So what I have here in the diagram, it was GDP when it came over, and it released energy,
Okay.
and there's energy there, which is activating this,
Okay.
and then the energy is found between the second and the third phosphate group, just like ATP.
Okay.
So now it's going back to GDP, because it lost the phosphate when it released the energy.
Okay.
So now it's GDP.
Okay.
And if we were to follow this further, it would no longer fit there, and it would come back as GDP. It would now be able to rebind with the alpha and the beta and the gamma subunits.
Okay.
Okay? So when it came over, it was originally GTP. That's why I have the phosphate over here. This is the last step. When it came over, so remember, let's see if I can. So when it came over, GTP was GDP, and then it uses the energy, and that becomes GDP, and it goes back.
Okay.
So it's cycling back and forth between those components.
Okay. And then one more question.
Yes.
So then why is the, okay, so then it says that, so then it releases the ATP, right? And then it becomes the.
So, well, ATP comes in and
Okay.
interacts with that,
Okay.
and forms cyclic AMP.
Oh, okay. Okay.
So ATP is another cellular molecule. It's diffusing around the cell. Without adenylate cyclase being active, ATP bumping into that, nothing would happen.
Okay.
But now that it's active, there's a site that recognizes ATP.
Okay.
And when it bumps into that site, again, totally random, then it's converted to cyclic AMP. But that only happens when this enzyme has been activated.
Okay.
Okay?
Thank you very much.
All right, so the second mechanism, second pathway by which a G protein can function is to provide calcium ions that function as the second messenger. So we have the same sequence where we have a hormone binding to a receptor. Okay, so we have this here. Hormone binds to the receptor. Again, the receptor itself is not an ionotropic receptor. It's not going to be a channel that opens. But we will see that effect. So the alpha subunit becomes activated with GTP. And now, instead of interacting with an enzyme present in the plasma membrane, it is going to interact with an ion channel. And that ion channel is a calcium channel. For extracellular calcium ions. When the alpha subunit interacts, and the GTP again releases energy and releases the same thing that we did with the adenylate cyclase. Instead of activating adenylate cyclase, the function is to open the calcium channel. So in the first example we used, the activity of the alpha subunit was to activate adenylate cyclase. In this case, the activity of the alpha subunit is to open the calcium channel. All right, rather than binding from the outside, like acetylcholine would do to its receptor, this binds from within the membrane. Once that is open, calcium ions enter the cell. And normally when we talk about an ion channel opening, it's the electrical current of the ions flowing into the cell that cause an event to occur. Right? Think of this more as within the muscle cell when sarcoplasmic reticular channels were open. And calcium exited the SR. What molecule did it bind to? Troponin. And troponins' function was to lock the tropomyosin in place on actin. In this situation, it binds to a similar molecule, calcium binding molecule, but it's known as calmodulin. So the second messenger is actually calmodulin plus the calcium. And as such, that now can diffuse throughout the cell. So the first case, I'll get in the habit here of writing it higher. What I need are the boards that flip up. Bless you. I wouldn't mark off if you put the calcium as a second messenger, because it does, but typically it binds to calmodulin, they both work together. Okay?
When a ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. The alpha subunit, with guanosine triphosphate attached, binds to the calcium ion channel, causing the calcium ion channel to open. Calcium ions diffuse into the cell and combine with calmodulin. The combination of calmodulin and calcium produces the response of the cell to the ligand. Phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the alpha subunit. The inactivated alpha subunit separates from the calcium ion channel and the channel closes. The alpha subunit recombines with the gamma and beta subunits, and the G protein recombines with the receptor.
Okay, yes?
Whenever this alpha subunit with GTP is bonded to a K, then it releases phosphate.
Well, that's how they put that at the end, but the conversion of GTP to GDP is what provides the energy to open this. Okay?
And it should happen right away.
Yeah. Pardon? When the G protein leaves, the alpha subunit leaves. Yes.
Alpha subunit.
Pardon?
So when would it leave?